Welcome to Chapter 3 and Anatomy and Research Methods. We're going to explore the human nervous system, the complex network of cells and circuits that makes everything we think, feel, and do possible. This isn't just anatomy for anatomy's sake. The nervous system is the foundation of psychology. It's the biological hardware behind our mental software. So let's walk through its structures, function, and organization. Why the nervous system matters. Imagine trying to drive a car without wires connecting the ignition to the engine. That's what the body would be without a nervous system. Disconnected parts with no coordination. The nervous system is the body's communication network. It receives information from the outside world, processes it, and sends commands to muscles and organs. Without it, there's no sensation, no movement, no thought. It's divided into two major parts. The CNS, which is the brain and spinal cord. This is the command center. It interprets, decides, and directs. The peripheral nervous system, or PNS, is all the nerves that extend beyond the brain and spinal cord, linking the CNS to the rest of the body. Think of the CNS as headquarters and the PNS as the field agents carrying orders and bringing back intelligence. The PNS itself splits into two functional systems. The somatic nervous system controls voluntary movement and transmits sensory information. When you raise your hand or type a word, that's the somatic system at work. The autonomic nervous system regulates involuntary functions, your heartbeat, digestion, and pupil dilation. And the autonomic system has two branches: sympathetic division, which activates fight or flight responses, increased heart rate, sweating, energy, and mobilization, and the parasympathetic division, which calms the body, slows heart rate, stimulates digestion, promotes relaxation. These two systems act like gas and brakes, keeping your body in balance. Running down the back is the spinal cord, a thick bundle of nerves connecting brain to body. Your dorsal roots bring sensory information in, ventral roots carry motor commands out. Gray matter in the center contains cell bodies. White matter around the edges contains axons carrying signals up and down. And don't forget, the spinal cord can act on its own through reflexes, like jerking your hand away from a hot stove. These bypass the brain to protect you instantly. Neurotransmitters in the ANS. The sympathetic system uses two key neurotransmitters: acetylcholine, ACH, used at the first in synapse, preganglionic to ganglionic neuron, and norepinephrine, NE, usually used at the second synapse, the ganglionic neuron, to target organs. In addition, the adrenal medulla releases epinephrine directly into the bloodstream, creating a body-wide surge of activation. That's why your heart races, your palms sweat, and your whole body feels on edge all at once. It can kick in everyday stress, like getting startled by a loud noise. When it goes too far, Kalat reminds us that prolonged sympathetic activation can be harmful. Chronic stress means the SNS stays on high alert. This can lead to high blood pressure, heart disease, digestive issues, and weakened immunity. What is the hindbrain? It sits at the very base of the brain, just above the spinal cord. It's sometimes called the reptilian brain because it controls core survival functions that we share with many other animals. Kalat Collat divides it into three main structures: medulla, pons, and cerebellum. The medulla oblongata is the lowest part of the brain, merging into the spinal cord. It regulates vital reflexes, breathing, heart rate, blood pressure, and vomiting, and it contains cranial nerves that control functions like swallowing and facial movements. Damage to the medulla can be fatal, which shows just how essential it is. Even though it doesn't think, it does keep us alive. Above the medulla is the pons, which literally means bridge. It acts as a bridge between the left and right hemisphere of the brain. It relays messages between the forebrain and the cerebellum. It plays a role in sleep and arousal. That is why damage to the pons can interfere with alertness or coordination. Cranial nerves. Unlike spinal nerves, which branch off the spinal cord, cranial nerves emerge directly from the brain, mostly from the brainstem. Humans have 12 pairs. They can be sensory, bringing information in, motor, sending commands out, or mixed, doing both. Together, they cover much of the head and neck and even reach down to organs like the heart and stomach. Collat emphasizes cranial nerves because they show the direct link between brain and body. They're essential in clinical diagnoses. Neurologists test cranial nerve function to identify brainstem damage, include both voluntary functions, chewing, facial expressions, and autonomic ones, heart rate, digestion, via the vagus nerve. Behind the pons sits the cerebellum, Latin for little brain. And although it's smaller than the cerebral cortex, it contains more neurons than the rest of the brain combined. Functions include coordination of movement, smooth, precise actions, balance and posture, motor learning, learning new skills like riding a bike or playing an instrument. In recent research, it's also linked to cognitive processes like attention, timing, and even aspects of language. Collat points out that damage to the cerebellum doesn't paralyze you, but it makes movement clumsy and uncoordinated, a condition called ataxia. The midbrain sits between the hindbrain and forebrain. Structurally, it's small compared to the sprawling cerebral cortex, but functionally, it's a relay center. Think of it as the middle manager, passing messages up to higher centers and sending commands back down to motor pathways. Collat highlights several major parts. The tectum, or roof, contains the superior colliculus, processes visual information, especially reflexive eye movements. It contains the inferior colliculus, processes auditory information, especially sound localization. Together, they help orient us to sudden sights and sounds in the environment. The tegmentum, or floor, is involved in movement, arousal, and reflex pathways. Houses part of the reticular formation, which regulates alertness and arousal. The substantia nigra is a dopamine-rich structure critical for movement control. Degeneration here leads to Parkinson's disease with tremors and rigidity. Red nucleus plays a role in motor coordination. It connects to pathways that help fine-tune movements. The forebrain, or prosencephalon, is the largest part of the brain, dominating in humans compared to other species. It includes the thalamus and hypothalamus, deep structures for relay and regulation, the limbic system, emotion and memory, the basal ganglia, movement and habit learning, the cerebral cortex, higher cognition, thought, and awareness. At the center sits the thalamus, which functions as the brain's hub for sensory information. Almost all sensory signals, except smell, pass through the thalamus before reaching the cortex. It acts like an air traffic controller, directing information to the right cortical area. Just below is the hypothalamus, small but powerful. It regulates hunger, thirst, temperature, and sexual behavior. It links the nervous system to the endocrine system via the pituitary gland, and it plays a role in emotions like fear and aggression. Kalat emphasizes the hypothalamus is about homeostasis, keeping the body in balance. The pituitary gland. It's a small pea-sized structure located just below the hypothalamus. It's often called the master gland. It controls hormone release throughout the body, but it doesn't act alone. The hypothalamus tells the pituitary what to do. The two parts are the anterior pituitary and the posterior pituitary. The anterior releases hormones like ACTH for stress, TSH for thyroid, LH, FSH for reproduction, and growth hormone. The posterior releases oxytocin, which helps in bonding, childbirth, and lactation, and vasopressin, which helps with water balance and blood pressure. Now let's shift to the basal ganglia, a group of subcortical nuclei buried deep within the forebrain. The main structures include the caudate nucleus, putamen, globus pallidus, and closely connected the substantia nigra in the midbrain. Functions include initiating movement. They help the cortex decide when and how to move, suppressing unwanted movements, preventing random or inappropriate motor actions, habit learning, turning repeated actions into automatic routines, and reward and motivation, dopamine pathways through the basal ganglia shape reinforcement learning. The basal forebrain is a group of structures located near the bottom and front of the forebrain, close to the hypothalamus. Key components include the nucleus basalis, the medial septal nuclei, and the diagonal band of Broca. Together, these nuclei send widespread projections throughout the cortex. The hippocampus sits in the medial temporal lobe, forming part of the limbic system. The name comes from the Greek word for seahorse because of its curved shape. It's closely connected to the amygdala, entorhinal cortex, and hypothalamus, forming circuits that link memory, emotion, and motivation. Kalat emphasizes that the hippocampus is not about storing all memories forever. It's about forming new memories and organizing them. The ventricles are a set of 4 interconnected cavities inside the brain. They're filled with cerebrospinal fluid, or CSF, which flows through the ventricles into the spinal cord and around the brain. The fluid is produced by the choroid plexus, a network of cells inside the ventricles. Think of the ventricles as reservoirs that keep the brain floating and cushioned. The meninges are 3 protective layers of tissue that cover the brain and spinal cord. From outermost to innermost, they are dura mater, the tough mother, arachnoid matter, the spiderweb layer, and pia mater, the gentle mother. These layers provide protection, stability, and support, while also creating space for the CSF to flow. The cerebral cortex is a thin, wrinkled sheet of gray matter covering the forebrain. It's only about 2 to 4 millimeters thick, but spread out it would cover about 2 square feet. The folds gyri, and sulci allow more neurons to fit into the limited space of the skull. It contains roughly 20 billion neurons and trillions of synapses, making it one of the most complex structures known in nature. The cortex is divided into two hemispheres, left and right, connected by the corpus callosum. Each hemisphere controls the opposite side of the body. Within each hemisphere are four lobes, each with specialized functions. The occipital lobe, vision, is located at the back of the brain. It's the primary visual cortex. Damage can cause blindness even if the eyes are intact. The parietal lobe is touch and spatial awareness. It's located at the top and back. It contains the somatosensory cortex, which maps sensations from the body. It helps with spatial processing, knowing where your body is in space. The temporal lobe, hearing and language, is located on the sides near the ears. The auditory cortex processes sound. Left temporal lobe is language comprehension, where Wernicke's area is, also important for face recognition and memory via the hippocampus. The frontal lobe is planning and control. It's located in the front. Contains the primary motor cortex for voluntary movement. The prefrontal cortex is crucial for planning, decision-making, impulse control, and personality. Damage here, as in the famous case of Phineas Gage, can dramatically alter behavior. Here are two regions that highlight just how versatile the frontal lobe is: the prefrontal cortex and precentral gyrus. The precentral gyrus is a fold of cortex located just in front of the cerebral sulcus. It's also called the primary motor cortex. Functions include direct control of voluntary movement. Each center of the precentral gyrus controls specific parts of the body. It's organized as a motor homunculus, a distorted map where areas with fine motor control, like the hands and face, occupy more cortical space than areas like the back or legs. It sends commands to the spinal cord and muscles. An example is when you decide to raise your right hand, the signal originates in the left precentral gyrus. Collat emphasizes the precentral gyrus is not about planning movement, but about executing it. At the very front of the frontal lobe lies the prefrontal cortex, one of the most evolutionarily advanced parts of the brain. It includes executive control, planning, decision-making, reasoning, working memory, holding information online while solving problems, inhibition, regulating impulses and emotions, personality and social behavior, guiding how we interact with others. Collat notes that damage here doesn't impair movement, but it profoundly changes personality and judgment. Classic case is Phineas Gage, who survived an iron rod passing through his prefrontal cortex. He was able to still move and speak, but became impulsive, irresponsible, and socially inappropriate. The precentral gyrus is the brain's motor output system, sending commands to move the body. The prefrontal cortex is the brain's executive system, guiding judgment, planning, and social behavior. Together, they show the frontal lobe's dual nature: action and reflection, impulse and restraint. A prefrontal lobotomy was a surgical procedure developed in the 1930s. Surgeons severed connections between the prefrontal cortex and the rest of the brain, especially the thalamus. The idea was to reduce symptoms of severe mental illness, for example, depression, schizophrenia, OCD, by calming overactive emotions. Portuguese neurologist Antonio Egas Moniz pioneered the technique in 1935, even winning a Nobel Prize for it. In the US, Dr. Walter Freeman popularized a faster version, the infamous ice pick lobotomy. In the 1930s and through 1950s, psychiatric hospitals were overcrowded and treatments were limited. Lobotomies appeared to reduce agitation and aggression. Families and doctors sometimes reported patients were calmer and easier to manage, but this calmness came at a terrible cost. Common effects included flattened affect, patients showed little emotion, loss of initiative, reduced motivation and goal pursuit, impaired decision-making, poor judgment and impulsivity, and personality changes. Many became passive, indifferent, or childlike. The procedure didn't cure mental illness. It often simply destroyed part of what made people themselves. The precentral gyrus controls voluntary movement. The prefrontal cortex plans, inhibits, and decides. The hippocampus organizes new memories. The basal ganglia shapes movement and habits. The thalamus relays sensory information. But none of these areas can function in isolation. The power of the brain comes from integration, networks linking these areas into dynamic systems. Think about this. When you look at a friend, you don't just see a blur of colors, shapes, and sounds. You experience them as a unified person. Their voice, their face, their expressions, and even your feelings about them all bind together. But how does the brain make this happen? The brain processing information in different regions. Occipital lobe handles vision. Temporal processes sound and language. Parietal manages touch and spatial awareness. The limbic system attaches emotion. Yet you don't experience these separately. Instead, they're integrated into a single coherent perception. That's the binding problem. How separate neural processes become one unified experience. So the takeaway, the binding problem asks how the brain combines signals from different regions into a unified experience. Current answers point to synchrony and attention, but the mystery isn't solved. What seems effortless, seeing a person, hearing their voice, feeling emotions about them all at once, is one of the deepest puzzles of neuroscience. Kalat emphasizes that understanding the nervous system isn't guesswork. It relies on research methods that let us look inside the skull, measure activity, and even alter brain function to test cause and effect. Structural methods show us what the brain looks like. CT scan uses X-rays to create cross-sectional images. An MRI uses magnetic fields to produce high-resolution images of brain structure. Brain damage can result from trauma, stroke, tumors, infectious, degenerative diseases. Each cause affects brain tissue differently. Sudden, localized damage versus gradual, widespread loss. Effects depend on location. The key principle, location matters. Whether it's in the occipital, temporal, parietal, frontal, depends on what the damage may be. Colott also stresses that damage isn't always the end of the story. The brain has plasticity. Surviving neurons can reorganize and take over some lost functions. Younger brains tend to recover better than older ones. Therapy and training can enhance memory. But plasticity does have limits. Some losses, like massive hippocampal damage, can be permanent. Studying brain damage has given us one of the most important discoveries in neuroscience. Broca's and Wernicke studies linked language to specific brain areas. Split brain research revealed hemispheric specialization. And modern lesion studies, combined with imaging, continue to map cognition into anatomy. Transcranial magnetic stimulation, or TMS, is a technique that lets scientists and clinicians influence brain activity without surgery or drugs. It uses a strong, rapidly changing magnetic field to induce electrical currents in targeted parts of the brain. A coil is placed on the scalp, and magnetic pulses stimulate neurons directly underneath. It's non-invasive, no electrodes inside the skull, no surgery required. Colott notes that TMS is especially valuable because it can create temporary, reversible lesions. By disrupting normal firing, TMS can turn off a brain area for a few seconds. An example is stimulating the occipital lobe can briefly disrupt vision. Excitation and other protocols, TMS can increase activity in the targeted region. This helps researchers test cause and effect. Is this brain area necessary for this function? Brain stimulation can be done in several ways. Electrical stimulation, TMS, DBS, optogenics in animals. Each approach highlights how neural circuits translate into movement, perception, and emotion. James Colott describes EEG as a window into the brain's electrical rhythms, a way of listening to the neural symphony as it plays out in real time. EEG measures the brain's electrical activity using electrodes placed on the scalp. Neurons communicate through tiny electrical signals, and when thousands fire together, their combined activity can be detected outside the skull. The EEG records these patterns as waves that change with state of consciousness and mental activity. Colott emphasizes that EEG doesn't tell us much about where activity happens, but it's excellent for showing when activity happens. It captures brain waves, event-related potentials, and abnormal activity. MEG measures the magnetic fields produced by electrical activity in neurons. It uses supersensitive detectors called SQUIDs. They pick up faint magnetic signals outside the skull. It shows by using excellent temporal resolution, timing, and better spatial resolution than EEG. It uses mapping brain functions before surgery. It's also used in studying timing of processes like language, perception, and attention, and in identifying seizure foci in epilepsy patients. The PET tracks metabolic activity in the brain by following a radioactive tracer, often attached to glucose. Active brain regions use more glucose. PET detects where the tracer accumulates, creating a color-coded map of activity. Good spatial resolution where activity is happening, but poor temporal resolution, changes unfold over minutes. It's used to identify brain areas involved in specific tasks. It's also used in studying neurotransmitter systems, dopamine in Parkinson, for example. fMRI uses the same technology as structural MRI, strong magnetic fields, and radio waves, but it focuses on function instead of just structure. It measures the bold signal, blood oxygen level dependent. Active neurons use more oxygen. Blood flow increases to those regions. The scanner detects those changes as a proxy for neural activity. So fMRI doesn't measure neurons firing directly, but it tracks the blood flow that supports them. Spatial resolution is excellent. It can localize activity down to a few milliliters. Temporal resolution, moderate. Seconds, not milliseconds, since blood flow changes more slowly than electrical signals. Output, color-coded images showing which areas light up during tasks. For example, when participants look at faces, the fusiform face area in the temporal lobe shows increased activity. Collat emphasizes that fMRI revolutionized brain research by allowing scientists to study the living, active brain. Phrenology was the 19th century attempt to link brain anatomy to behavior by examining the bumps on people's skulls. It turned out to be wrong in method, but it left a surprising legacy in shaping how we think about the brain today. It was developed by Franz Gall in the 1800s. It was wrong because skull shape does not reflect brain anatomy. Personality cannot be read from bumps. Phrenology lacked scientific testing. Now we'll look at modern methods of correlating brain anatomy with behavior. Two powerful ways to look at the brain structure, the CAT scan and MRI. The CAT scan uses x-rays taken from many angles. A computer combines them into cross-sectional images of the brain. It shows general brain structure, tumors, bleeding, swelling, and skull fractures. The strength is that it's quick and widely available, and it's good for emergencies. Limits are lower resolution than an MRI, and it uses radiation exposure, so less ideal for repeated use. After a head trauma, a CAT scan is often the first test to quickly check for bleeding. MRI uses strong magnetic fields and radio waves to align hydrogen atoms in the brain. When the atoms return to normal, they release signals that form detailed images. It shows high-resolution images of gray and white matter, small structures, and abnormalities. It's sharper and more detailed than a CAT scan, but it's more expensive and it's slower. So there you have it. Brain size is not correlated with higher intelligence according to research. IQ is actually correlated with the amount of gray matter. Collat reminds us that the answer about whether men and women brain size intelligence should be compared, that the answer requires nuance, because while biology shows some differences, intelligence is far more complex than raw brain measurements. On average, men have larger brains than women. The difference mostly reflects body size, since men tend to be larger overall. But bigger doesn't mean smarter. Elephants and whales prove that. The important factor is efficiency of organization, not just volume. Despite size differences, research consistently shows no overall gender difference in IQ. Men and women on average score about the same on general intelligence test. Where differences sometimes appear is in specific abilities. Men slightly stronger in some spatial and mathematical reasoning tasks. Women slightly stronger in verbal fluency and memory for object locations. But these differences are small and influenced by culture, education, and experience. Collott emphasizes that men and women's brains may be organized differently. The takeaway: men, larger brains on average, but not more intelligence. Women, smaller brains on average, but equally intelligence. See you next time for Chapter 4.